all, welcome to my video seminar presentation. Myself Salma, Sakkariya, student of Electronics and Biomedical Engineering. So, my topic is enhancing wound healing via circadian controlled melatonin and phototherapy. This is the roadmap for my presentation. It begins with an abstract introduction, literature survey, objectives, then move on to the circadian rhythm and wound healing, melatonin in wound healing, phototherapy in wound healing, integration and future direction, then applications, challenges and limitation, conclusion, future scope and references. Then begins with the abstract. It begins with an overview of circadian biology in wound healing, then move on to the therapeutic role of melatonin, then analyzes the phototherapy approaches, then highlight the integration of melatonin with phototherapy as a synergistic and circadian control strategy for improved healing, then concludes with the future perspective, challenges and application of integrated therapy. Let's move on to the introduction. Circadian rhythm is the 24-hour biological clock in our body that regulates physical, mental, and behavioral process. It influences cell proliferation and migration, angiogenesis, collagen remodeling. The first picture shows the circadian rhythm of a nocturnal animal. As we can see that several cells are increased at daytime and some of them are increased at high time. Melatonin release is high at night time. Then melatonin, a natural hormone produced by the pineal gland in the brain. It is also called sleep hormone because it regulates circadian rhythm. Then secretion is less during day and high during night. The second picture shows the picture of the cell proliferation in the diabetic rat when it is treated with the melatonin. Then we can move on to the phototherapy. Uh, the picture shows the difference between blue light phototherapy and red light phototherapy. At blue light is used for infectious treatment and red light is used for depth treatment. Then move on to the literature survey. The first paper I took is cutaneous photoregenerating of light emitting diodes via the melatonin membrane receptor pathway, which shows the photobiomodulation studies. And the second paper is Janus inspired core shell structure hydrogel programmatically releases melatonin for reconstruction of post-operative bone tumor, which shows a programmable core shell hydrogel was designed to release melatonin in stages, which shows the dressing of a wound healing treatment. Then the third paper I took is melatonin and bacterial cellulose regulate the expression of inflammatory cytokines VEGF, PCNA and collagen in cutaneous wound healing in diabetic rats. Then this paper shows the diabetic rats wound healing using melatonin. Then the fourth paper is impact of blue light therapy on wound healing in impact of blue light therapy. This paper shows how blue light is impacted in a treatment. Then the fifth paper I took is injectable light responsive hydrogel dressing promotes diabetic wound healing by enhancing wound angiogenesis and inhibiting inflammation. This paper shows the hydrogel dressing that promotes the wound healing of a diabetic patients. So this is the objectives of my seminar. The first objective is the overview of circadian rhythm and wound healing. Then the second one is importance of melatonin in wound healing. Then the third one is role of phototherapy in wound healing. And the fourth is the integration of circadian, melatonin and the phototherapy. So let's move on to the first objective, circadian rhythm and wound healing. Circadian rhythm is the 24-hour biological clock in our body that regulates physical, mental and behavioral process. Circadian rhythms regulate multiple tissue repair and regeneration processes across a 24-hour cycle. As we can see in the figure, the first portion shows the daytime and the next portion shows the nighttime. It shows the graph of a nocturnal animal in which its skin wound closure is faster in night and slower in daytime. Similar, like the gastrointestinal regeneration is high in day and slow in night. And as we can see that the melatonin production is high in night. Melatonin secretion and neuroendocrine signals synchronize these processes. In skin, wound closure and fibroblast activity are faster during the active phase compared to the rest phase, and wound healing is faster during rest phase. This slide shows the role of clock genes in healing. This shows that it operates through a feedback loop of core genes. The clock genes are clock, BML1, PR, CRY, ROR, REV, etc. As we can see in the first, the 00 shows that the transcriptional activation, which means nuclear phase, clock and BML1 form a heterodimer that binds DNA at E-box promoter elements. E-box is the short DNA sequence found in the promoter region of many genes. As we can see, clock BML1 activates transcription of PER and CRY. Then the second, that is at 6, the translation and cytoplasmic accumulation is take place. Then the first process takes place in the sixth portion is PER and CRY mRNAs are translated in the cytoplasm. Then their proteins accumulate. Then these proteins initially remain cytoplasmic and undergo post-translational modification. Then move on to the next portion, that is 12, the complex formation, phosphorylation and timing control. That means PER and CRY added together and form a combination of PER CRY. Then moves to the 18th portion. It shows the nuclear entry and repression. PR cry complexes translocate into the nucleus and inhibit clock BML1 activity. Then the loop is closed as transcription is on. In the late night, early morning degradation it is happen. If this clock is disrupted, slower embed own healing is happen. This is the total clock gene function of a wound healing phase. If any disruption is happened in these clock genes, it will severely affect in the wound healing process. This slide shows the circadian timing of repair. The circadian clock regulates cell cycle timing, redox balance, and DNA repair efficiency. As we can see in the figure, that is normal clock, time restricted feeding shifted clock, and DML1 minus clock. Under a normal clock, ROS damage is minimized, enabling optimal stem cell and tissue repair. Then the time restricted feeding shifted clock, it has increased ROS, impairing DNA repair. Then in DML1 clock gene is not present, that is DNA damage is high and stem cell dysfunction is present. Then move on to the next slide, circadian regulation of stem cell. This figure shows that how circadian is regulated in the stem cells. Epidermal stem cells drive reepithelialization during wound repair. Their proliferation and differentiation are regulated by circadian genes, that is BML1 and PER. Then disruption of circadian control reduces cell proliferation and impaired repair. As we can see, the first image is hair follicle in early anagen, and the second picture is endofollicular epidermis. That means the structure of a stem cell. The hierarchical model shows that it is like a that means a company CEO and the worker relation, and the next the stochastic model shows a flat like organization, which means any cell can access to the epidermal stem cells. So move on to the next is the case study of role of PER2 in wound healing. As we can see in the figure, the first shows a control myofibroblast, then the PER2 KO myofibroblast. That means a rise. That means a mice is treated with a normal treatment, and PER2 KO means treated with PER2 KO. As we can see, the red substance in the figure is the myofibroblast. In the second picture, that is treated with PER2, the myofibroblast is arranged in structured way and it helps to treat the wound efficiently. In this figure, we can see in day 6, in control, collagen fibers are starting to reappear in the wounded, early deposition and alignment. In PER, it is delayed ECM deposition. Then we can see on the day 10, scar tissue form well aligned collagen fibers. Then PER2 KA collagen deposit is disorganized. Control mice display organized tissue remodeling and faster wound closure. PER2 KO mice exhibit disrupted collagen organization and delayed wound repair. Highlights the role of circadian clock and gene PER2 in the remodeling of tissue regeneration. Then the next picture shows that PER2 controls ECM remodeling by regulating periostein deposition. In control mice, periostein is well organized, supporting fibroblast migration and tissue repair. In PER2 KO, mice periostein distribution is disorganized, leading to delayed reepithelialization and poor wound closure. As we can see that it confirms the circadian role of PER2 in coordinating the ECM remodelization and structural repair mechanism. We can move to the summary of circadian influence in wound healing. Wound healing is time of day depend faster during the active phase, slower during rest. Circadian clock genes regulate fibroblast migration, keratinocyte proliferation, and stem cell activation. The central brain clock coordinates with the peripheral clocks in skin and immune cells to regulate repair processes, disruption of circadian rhythm, delayed wound closure, and pure tissue organization. Then, and this slide shows the summary of how clock genes works in the efficient wound healing. Uh, then move on to the second objective, melatonin in wound healing. Melatonin is a natural hormone that is secreted by the brain. Melatonin is also called N-acetyl-5-methoxytryptamine. It is the primary circadian hormone secreted by the pineal gland. Secretion follows a circadian rhythm. Low in daylight, peaks at night, serving as a biological time cue. As we can see in the figure, melatonin secretion is high in the night time and low in the daytime. Then this slide shows the biological roles of melatonin. Antioxidant. It is act as an antioxidant that is scavenges ROS, reduces oxidative stress, protects DNA and proteins. Then also it acts as an anti-inflammatory that is downregulates pro-inflammatory cytokines and accelerates inflammatory resolution. Then it also acts as angiogenic and pro-regenerative. It means enhances endothelial migration, tube formation and VEGF signaling. Then it also helps in the matrix remodeling, promotes fibroblast proliferation, upregulates PCNA and collagen 1 synthesis, improving granulation and reepithelialization. And this figure shows how these biological roles are activated through the melatonin. Then the next slide shows the role of melatonin exosome. Melatonin exosome is also called MT-exo, which is applied to the diabetic wounds, and the mechanism shows in the figures are the PTEN is high and the AKT phosphorylation is low. That means macrophages are shift from M1 to the M2. Then the effect is inflammation is low and the angiogenesis is high and the collagen deposition is also high. That means the final outcome is faster wound closure with the improved tissue regeneration. Then this is the fabrication of a melatonin hydrogel nanocarrier. As we can see that it is a Janus inspired core shell hydrogel. It improves wound closure rates with the combined with the free melatonin delivery. That means first a gel is molded into a molding cast. Then after it is molded, then it is also put into another mold and it also produces small compartment for storing the melatonin. This is the spectrum graph showing how melatonin gel works in the wound. Then this is the graph that showing the fabrication of melatonin carriers, that is design, control, release and the biological effects. As we can see that controlled release profiles, and this next graph is showing the in vivo wound closure. How this release is, how this drug release is compared to the wound closure. That this graph is shows here. As we can see that by using the normal treatment and also using the melatonin treatment is efficient in wound closure. Then this is the next fabrication, that is nanofiber biomaterial scaffold. The first picture shows that blend electrospinning. That means the solution is blended and it is given to the molder. Then the second is coaxial electrospinning. That means two solution is added, that is sheath solution and core solution. Then a core sheath structure is formed. Then the C is emulsion electrospinning, that is emulsion liquid is poured into the spinner and a core sheet structure also formed. Then the next is side by side electrospinning, that means two different gel can be obtained. The last one is the most efficient method. Then move on to the next. This also shows that how these electrospinning nanofiber act in the wound closure. This is the picture of a wound treatment done in the mice. As we can see that the therapeutic efficiency and nanofibers was highly efficient. This slide shows the case study of melatonin plus bacterial cellulose, that is diabetic wound healing. This is the result of a diabetic wound healing in a rat. That is, melatonin and bacterial cellulose combined together. As we can see in the final picture, the structures are improved, and that means the cell proliferation is high. That means the wound closure is better when treated with melatonin and bacterial cellulose. Then this slide shows the summary of melatonin effects in wound healing. As we can see that there are pictures from A to H. Each picture shows the different amount of melatonin is treated with the different images of cold fish. As we can see at the final, 0.1 gram of melatonin is given, and it is highly efficient. Melatonin plays a multifaceted role, that is antioxidant, anti-inflammatory, pro-angiogenic, pro-collagen, supports EPC survival, fibroblast proliferation, macrophage polarization, delivers C systems, nanofibers, enhances therapeutic effects. Then move on to the third objective, phototherapy in wound healing. As we can see that the first picture shows the light spectrum for phototherapy. Photobiomodulation uses visible to NIR lights. The blue light is used for antimicrobial superficial penetration. Then the red light is used for pro-healing stimulates fibroblast. Then the NIR is used for deeper tissue penetration. This slide shows the comparison of blue versus red light. As we can see that in the B picture, a rat is treated with the blue and red light. When treated with blue light, the scar is still there, but it is treated also. But as we can see in the red light treatment, the scar is completely removed, and small circle shaped dot we can see there. So, red light is more efficient in deeper tissue penetration, and the blue light is used for antimicrobial infection treatment. This shows the fabrication of light responsive hydrogel. That means how a wound dressing is fabricated with light responsive gel, and the result is shown that it is highly effective in wound closure and wound closure and scarring treatment. So this is the case study, photobiomodulation in diabetic wounds, a typical progression of wound healing in untreated control animals and of wound treated with pulsed LED photobiomodulation with red, green or blue light respectively. As we can see that from day 0 to day 24, in red light this scar is almost completely removed, and in another scar is present somewhat, and as we can see that in control mice there is no improvement in this scar. So healing rate is calculated using a equation a1 minus a2 by p1 plus p2 into 0.5, then divided by t2 minus t1. This slide shows the case study of photobiomodulation in pediatric skin injuries. This is the picture of a 12-year-old boy hospitalized for respiratory failure, and he got this injury by regularly using the respiratory mask. The first picture shows the first day of the injury, and the last picture is completely healed wound by using the photobiomodulation treatment. Then move on to the summary. Blue light is the antibacterial, enhances fibroblast migration and reduces scar formation. Then red light is used for stimulates angiogenesis, fibroblast proliferation, collagen deposition. Then NIR is used for deep tissue penetration, improves mitochondrial function and circulation. Melatonin plus LED synergy. Light activates MT1 and MT2 receptors, boosting collagen remodeling and tissue regeneration. The clinical benefits are faster wound closure, reduced infection, improved scar quality. Challenges are dose optimization, penetration depth, limits patient variability. Then move on to the fourth objective, integration of melatonin and phototherapy. Why integration matters means dual mechanisms energy. That is, melatonin acts as antioxidant, anti-inflammatory and pro-angiogenic. Then phototherapy helps in the stimulates mitochondrial activity, collagen remodeling and antibacterial effect. When it is melatonin and phototherapy is combined together, the complementary and amplified wound healing response is getting. And the circadian alignment is the major advantage when we are integrating both these treatment. That is, melatonin regulates circadian rhythm, while phototherapy can entrain biological clocks. Integration ensures therapy delivered at optimal biological time, accelerating tissue regeneration. Then controlled and targeted healing. Light responsive biomaterial release melatonin on melatonin on the damaged skin. Then localized therapy reduces systematic side effects. Then as we can see that this is the picture of a histological Mason trichome stained photomicrographs of treatment groups at the end of 1 week. This is the completely whole treatment happened within the 1 week. As we can see that by using melatonin phototherapy and aligned with the circadian rhythm, the wound is completely healed and the structures are visibly structurally arranged. Then advantages of integration are chronosynergistic therapy aligns treatment with the body's natural circadian peaks in cell proliferation, angiogenesis and repair, enhanced efficiency, melatonin release and PBM light exposure timed to night cycle maximize antioxidant and regenerative effects, reduced side effects. Circadian based delivery minimizes oxidative stress and inflammation during vulnerable phases. Optimized healing phases. Early night melatonin decreases inflammation later. Photobiomodulation increases angiogenesis and collagen, matching wound healing stages. Then next advantage is personalized medicine. Enables patient-specific treatment schedules based on circadian chronotype. Then the applications are chronic wounds, pressure ulcers, venous ulcers. Next one is diabetic wounds, impaired angiogenesis and oxidative stress. Third one is infected wounds, staphylococcus and biofilm targeted nanocarriers. Then the fourth one is post-surgical repair, faster recovery and scar quality. This slide shows the challenges and limitation of integrated therapy. The first one shows that clinical translation. That means clinical trial can be somewhat difficult for trial in a human. Then second one is delivery issues. The delivery of melatonin is somewhat difficult, and the dosing and timing. Every wound and every human body is different, and everyone's dose and timing should be different. And the cost and accessibility, then safety aspects, then patient variability. Then these are the result occurred through the melatonin treatment, phototherapy treatment, and the integrated therapy. All these results are combined in a graph and shown in the next slide. And this is the comparative outcome of melatonin, photobiomodulation, and the integrated therapy. As we can see, in all the parameters such as oxidative stress, inflammation, angiogenesis, collagen deposition, wound closure speed, infection control, and circadian alignment, the integrated treatment is high, and we can understand the efficiency of integrated treatment. Then we move on to the conclusion. Circadian rhythms critically influences wound repair with studies showing that healing outcomes vary depending on the time of day, highlighting the need for chrono-aligned therapies. And melatonin supports healing via antioxidant and anti-inflammatory actions with biomaterial carriers enabling sustained release. Phototherapy promotes antimicrobial defense, angiogenesis, and collagen deposition enhanced by light-responsive biomaterials. In integrated melatonin phototherapy approaches, provided angiogenesis circadian controlled healing with a strong translation. That means healing is improved when integrated phototherapy and melatonin treatment is occurred. And future scope, personalized therapy explores circadian based dosing and light exposure schedules tailored to patient chronotypes. Then infection control, integrate antimicrobial nanocarriers with melatonin and light therapies for multi-drug resistant wound infection. Then regenerative medicine combine melatonin and PBM with stem cell or exosome therapies for complex tissue regeneration. Smart biomaterials develop next generation injectable and light responsive hydrogens with programmable phase specific release. Then exudate collection system. That means that in some wound there is lot of exudates are there. Then we can connect with the exudate tube, and it will be go to the exudate microbial compartment, and it will be a better advantage in a smart wound dressing. Then these are my references, and that's all. Thank you.